Bioaccumulation. Biosorption is the term which is used to describe the ability of microorganisms or biological materials such as plants or plant-derived biomass to bind and concentrate heavy metals and other pollutants like azo-dyes and hydrocarbons from their surroundings. Among its various mechanisms, bioaccumulation is one of the processes which is biologically active process involving the uptake of contaminants into living cells. Unlike passive mechanisms such as surface adsorption, bioaccumulation requires involvement of energy and metabolically active microorganisms that can transport and store pollutants inside their cells. Bioaccumulation refers to the gradual accumulation of substances such as heavy metals and other pollutants in an organism within the time. This process occurs when the uptake rate of substance exceeds its rate of elimination. As the elimination is occurring very slowly, the absorbed material gets accumulated inside the cell. As the accumulation is occurring inside the biological entity, it is called as bioaccumulation. While bioaccumulation can occur at the whole organism level, it is fundamentally governed by intracellular mechanisms that determine how pollutants are processed once internalized by the cell. Upon cellular entry, the metal ions and other toxicants can cause significant biochemical disruption. Therefore, organisms have evolved intracellular detoxification mechanisms to tolerate the toxicity while still allowing these substances to accumulate inside the cell. That means when these pollutants are taken up by certain organisms, these pollutants can harm the organism itself. It can kill the cell. So, as a strategy of survival, organisms have develop different mechanisms which help the organism to survive in presence of these toxins while these pollutants are getting accumulated inside the cell. These mechanisms not only protect the cells from acute damage but also influence the magnitude and persistence of bioaccumulated pollutants. This process is important as heavy metals and organic pollutants pose a significant threat to ecosystem and human health due to their toxicity, persistence, and non-biodegradability. That means they get accumulated in the nature, they can enter the food chain and enter human systems including reproductive system, immune system, and some of these pollutants may cause cancer. They are termed as carcinogenic or they might show teratogenicity. That means they can harm the growing fetus or they may be mutagenic. Traditional removal methods are often costly, energy intensive, and generate hazardous sludge. The traditional methods mainly include chemical methods and physical methods. Biosorption is a more recent method which presents a greener alternative which is eco-friendly. It will produce less harmful byproducts, and bioaccumulation in particular offers advantages such as selectivity and higher uptake capacity. Since it is an active, metabolism dependent process, bioaccumulation is particularly effective in environments with low pollutant concentrations, where passive mechanisms may fail. Understanding this mechanism is vital for developing efficient, sustainable bioremediation technologies. This is one of the methods where the pollutants are taken up against the concentration gradient. That means even if the metals or other pollutants are getting accumulated inside the cell, that means the concentration of these pollutants is increasing in the cell. It is more, and sometimes it might be more as compared to the surrounding environment. So in such cases also, the pollutants from the environment are taken up by the cell, and for this there is need of energy in form of ATP molecules. So this is the specialized process where other processes like passive adsorption, diffusion can occur only among the concentration gradient without any involvement of energy. Bioaccumulation is the process by which living organisms absorb and concentrate substances from their environment into their cells over the time. It can occur through direct uptake from surrounding polluted water or soil via membrane transport system. In microbes, bioaccumulation may involve ATP-dependent transporters that move metal ions across the active cell wall into the cytoplasm. Once inside, these substances may be detoxified or stored using metal binding proteins which are called as metalloproteins, or by compartmentalization into organelles or vacuoles where they will become unavailable in the cytoplasm. Unlike biosorption, via surface adsorption, bioaccumulation is selective, dynamic, and dependent on the organism's metabolic state. If the organism is metabolically active, the rate of bioaccumulation will be more. The intracellular mechanisms involve chelation, compartmentalization, and then detoxification. Once a pollutant enters the cell, it must be detoxified or sequestered to avoid cellular damage. If these pollutants remain in their original form inside the cell, they can harm the organism, so the process becomes ineffective. So, for survival, the organism will make the molecule less harmful. Several intracellular mechanisms are involved in this process. First is chelation. It is one common strategy where the metal binds to ligands such as glutathione or metallothioneins. Another mechanism is compartmentalization, where these pollutants are sequestered in vacuoles or granules to reduce their bioavailability within the cytoplasm. Enzymes may also reduce the metal's toxicity by altering its oxidation state, for example, reduction of chromium plus 6 to chromium plus 3. These mechanisms not only protect the organism but also contribute to long-term metal retention, so that metal or any other pollutant which is taken up by the organism will remain in the accumulated state inside the microbial cell and it will be sequestered from the environment. Chelation and stable storage. Chelation plays a central role in both detoxification and bioaccumulation. Intracellular chelators like glutathione or metallothioneins bind tightly to the metal ions. This binding results in formation of stable complexes which are biologically inert complexes. So these complexes cannot interact with the other cell organelles or it cannot interfere with the cell's metabolic pathways. These complexes prevent the metal from interacting with vital cellular targets, thereby reducing their toxicity. However, because the chelated metal remains within the cell, it contributes to the overall bioaccumulated burden. This mechanism allows for long-term intracellular retention of metals in detoxified form. That means the metal will remain inside the cell but it cannot harm the cell. Compartmentalization and bioavailability control. Subcellular compartmentalization is another mechanism which contributes to bioaccumulation. Metals may be actively transported into vacuoles, lysosomes, or precipitated into insoluble granules, thereby isolating them from sensitive areas of the cytoplasm. While this spatial segregation reduces metal bioavailability and toxicity, it will not remove the metal or any other pollutant taken up by the cell from the organism. That means these pollutants will get accumulated inside the cell in separate compartments. Instead, uh, as these pollutants are not getting removed from the cell, they will result in internal sequestration, effectively increasing intracellular metal loads and contributing to persistent bioaccumulation at the cellular levels. Third is enzymatic transformation and retention. This is more effective mechanism as due to certain enzymatic activities inside the microbial cell, the pollutant is converted to less toxic form. That means the pollutant gets transformed into less harmful form, though it will remain inside the cell only. Enzymatic mechanisms can transform metals into less toxic or less mobile forms. For example, the intracellular reduction of chromium plus 6 oxidation state to chromium plus 3 oxidation state decreases the metal's reactivity and toxicity. However, chromium plus 3 has a higher tendency to form insoluble complexes, and hence it will remain in the insoluble complex form inside the cell, thus enhancing the bioaccumulative potential. Enzymatic reduction, while protective in the short term, can therefore facilitate long-term intracellular metal persistence. So, in conclusion, we can say that intracellular mechanisms such as chelation, compartmentalization, and enzymatic transformation play dual roles. They detoxify the pollutants and simultaneously enable their retention within cells. As a result, these processes are key determinants of an organism's capacity to accumulate metals or other pollutants over time. Bioaccumulation process makes the pollutant unavailable in the environment. Factors that affect bioaccumulation include several environmental factors as well as microbial factors. It includes the species of microorganisms, activity of the microorganisms, and environmental conditions such as pH, temperature, and the presence of pollutants. The nature of the contaminant, whether it is an essential or non essential element, also plays a role, as essential metals like zinc or copper may be more readily taken up as they are required by the organism itself for its metabolic activities. Additionally, nutrient availability, stress response, and the presence of specific transport proteins in the cell membrane can significantly affect bioaccumulation efficiency. If nutrients are available in abundance, organisms will grow rapidly. Also, there the biomass increases. All these factors result in more bioaccumulation. If the organism is capable of stress responses, that means if it is capable of surviving in presence of toxic pollutants like heavy metals or azides, then due to the higher survival rates, organism can show bioaccumulation at higher rates. And also, the organism should possess specific transport proteins which are required to transport these pollutants from outside the cell to inside the cell. Optimizing these factors is the key to improve pollutant removal in real world applications. Microorganisms involved in the process of bioaccumulation include bacteria such as Pseudomonas aeruginosa, Bacillus subtilis, Ralstonia metallidurans, and there are some genera of fungi like Aspergillus and yeast like Saccharomyces cerevisiae. All these microorganisms show effective bioaccumulation. These organisms use specialized metal transport systems to take up and detoxify harmful elements. Some produce intracellular proteins like metallothioneins that tightly bind and store heavy metals. Others convert the metals into less toxic forms via enzymatic processes. These enzymatic processes allow the organisms to survive and even thrive in contaminated environments. Their ability to actively uptake and store pollutants make them valuable agents in bioremediation strategies. Applications: bioaccumulation based biosorption has wide ranging environmental applications. It is being used in wastewater treatment to remove trace metals like cadmium, chromium, mercury, and lead, particularly at low concentrations where passive biosorption is less effective. It is also useful in bioremediation of contaminated soils, phytoremediation, and biomining, where metal accumulating microbes or plants help to extract valuable elements. Furthermore, genetically engineered microbes with enhanced uptake capabilities are being explored for selective metal recovery in recycling and resource recovery systems. Bioaccumulation offers a promising route for sustainable and selective pollutant management.